and everyone online. I'm very, very happy today to introduce these talks. Usually we have talks in the morning, but we adjusted so that Vicky and Al traveling from Australia could reach us on time and not too jet lagged, I hope. But it is my pleasure to introduce you today to two speakers from the Department of Primary Industries, working for the government in New South Wales, Port Stephens. And the first speaker is Alistair Becker, a senior research scientist, and then followed up by another presentation. I'll give it to you just now. Thank you, Al.
Cheers.
Let's see. Yeah.
Okay, I will point out that it's actually midnight in Australia right now, and we've been in the country for not even 24 hours, so hopefully I can make some sense with what I'm going to talk about today. But yeah, so I'm a scientist with Fisheries in New South Wales, and my job at Fisheries is essentially to run and manage, if you like, the research and monitoring that we do in support of what we call the Fisheries Enhancement Program. So with today's presentation, I'm going to give you a bit of an overview of the Fisheries Enhancement Program, the work that we do there, because it might be perhaps maybe a little bit different to some of the ways you do things here in South Africa. But yeah, essentially, I'm not going to sort of go into the nitty gritty of any particular sort of scientific study, more give you a bit of an overview of what we do as a whole and how our research and monitoring supports that. So you're probably thinking, what is Fisheries Enhancement? Well, essentially what we're looking at doing is increasing local fisheries productivity. So when I say local, I've got that sort of estuary scale or a sort of a small section of coast through management actions such as deploying artificial reefs or stocking fish into estuaries. I put this slide up at all my presentations. There's a bit of a caveat at the start because I get a bit nervous about some of the work that we do and the way some people sort of may perceive that. But essentially, the enhancement program is really like an additional lever for fisheries managers. Okay, so it's certainly not, say, the first tool in the toolbox that the managers in the state go to when there's like a concern about I don't know, like say mulloway or cob, as you guys call them. We don't just go, well, don't worry about that. We'll just grow some fish and stock them and that will solve all of our problems. By far and large, our fisheries are managed through, like, I guess, more traditional type approaches. So input and output controls, so bag limits, size limits, quotas for commercial managers, input controls, which is probably more aimed at the commercial sector. We do a lot of habitat restoration, which Vicky will talk about, spatial management, all those kind of things all come into the way we manage our fisheries in New South Wales. And this enhancement program is simply there as an additional tool that we can use. It's also entirely funded by recreational fishing license fees. So if you want to go fishing in New South Wales, you have to buy a license. That money goes into a kitty, which then funds all sorts of different things, including the entire enhancement program. So because of that, obviously, there's a focus of our work towards the rec fishing sector. So, for example, all of our stocking work is done into estuaries that we call recreational fishing havens, where commercial fishing is actually banned. So in terms of, I guess, the scope of the program, in terms of the artificial reefs, there's now 11 reefs deployed along the coast of New South Wales. It's probably not particularly helpful, this map, because there's no scale on it, but it's about 1,000 kilometres of coastline from north to south. And you can see these blue dots here is where we have those offshore artificial reefs. They're pretty big. That's one of them over there. Some are made from steel, some are made from concrete. They're super expensive. Yeah, as I said, we sort of have them along the coast. We have 6 estuarine artificial reefs as well, which I'm not going to go into today, but we do have them in a bunch of estuaries dotted along the coast as well. And then obviously the second component to the enhancement work that we do is the marine, we call it marine stocking, but it's actually stocking into estuaries. And since 2015, we've stocked 4 species, eastern king prawn, which is like a pinnate prawn, yellowtail kingfish, which you got karanga, I think you guys call them yellowtail. And then the final 2 are what we're stocking at the moment, and we've been doing that since 2021, which are dusky flathead, which I think you call estuary gurnard. Bar-tailed flathead. Yeah, okay, similar, yeah. This species is actually endemic to Australia, but it gets big. It gets like 1.2 metres, so it's a large platycephalidae. And then mulloway, which you guys know is cob. So they're the 2 species that we've been stocking since 2021, and it's probably going to be the 2 species which we'll focus on most likely over the next few years. Yeah, and that's where it's sort of going. And again, probably doesn't mean too much to you, but so as I said, I kind of like lead and coordinate the research and monitoring that we do in support of that program. So, I mean, the main reason for that is to ensure that the program is meeting its goals and its objectives. And obviously, you can't tell if a program's meeting its objectives if you're not monitoring something. Importantly, it allows managers to alter their practices over time if a particular management action isn't having the desired outcome. Okay, so ideally, and this has happened, the data that we're producing is sort of demonstrating that a particular management action really isn't having the desired effect, and then those practices have been altered over time. It minimises ecological risk, and certainly there's ecological risk associated both with deploying artificial reefs, but probably more so with stocking fish. And finally, it provides an opportunity for stakeholders to be directly involved in the program, which is a really critical aspect for us. Like as a government department, we need to obviously engage with our stakeholders. That's a really important aspect of the work that we do. So we have like a growing citizen science program where we have recreational anglers out there in the estuaries that we're stocking. They're taking fin clips of the species that we're stocking. And then we then use like genetics or parentage analysis to determine whether the fish they're catching are stocked ones or wild ones. That's been a fantastic extension tool for us, as I said, as a government department. So in terms of what we do, I guess some methods and approaches, I sort of split this up into the reefs and the stocking. We've done a lot of for the reefs, a lot of underwater video work, so stereo bruvs, bruvs are underwater videos. We look at the species compositions around the artificial reefs, how they change over time. Obviously, we look for the presence of recreationally important species, remembering all this stuff is funded by people's fishing license fees. So we want to ensure that there's at least something there that people are hoping would like to catch. We've done a lot of acoustic telemetry work on the artificial reefs, which is basically just tracking fish around. We put little transmitters inside them and surgically implant them. And then we track their movements around the reef. So this has been used to look at the way fish use structure, productivity estimates, looking at how long they spend there, and interactions with threatened species, which I'll talk about at the end of the presentation, so I won't get into that too much right now. And we've done a lot of hydroacoustic surveys. So these are basically echo sounder surveys where we run basically transects backwards and forwards over the artificial reefs, and we map the density and the distribution of small pelagic fish around the structures, and again link this to trophic interactions. And we've done some pretty neat work combining some of the hydroacoustic work with some of the acoustic telemetry to demonstrate linkages between, say, pelagic food resources and benthic fish that are sort of utilizing that reef as structure. For the marine stocking, again, we do a lot of acoustic telemetry work. In this case, looking at the habitat use of the fish that we're stocking, particularly for the juveniles, because we know that if we can stock fish directly into a key juvenile habitat, they've got a much better chance of surviving. So we've done a lot of work, and again, I'll talk about a case study where we've done this, where we basically grow fish up in a hatchery, release them with acoustic transmitters, and then identify the kind of habitats that they're using, and then use that to guide our stocking practices. We look at the growth and the contribution of stocked fish to the fishery, and again, this is where that citizen science project really sort of comes into it. Where we have those anglers out there catching fish, and we use the genetics to identify them. And we back this up with independent fisheries surveys as well. So I actually had a team out last week doing some surveys in a couple of the estuaries. And we do a lot of genetic work too. Now, I understand people at SAIAB are probably pretty well versed in genetics, but we look at the genetics of the brood fish that we're using. We only use wild brood fish for our stocking programs. We look at the genetics of the fish leaving the hatchery. We look at the genetics of the population structure of the species that we're stocking, because you've got to remember that we're stocking these species and they're able to obviously breed with their wild conspecifics. So that's a critical aspect of our stocking program that we need to keep them atop of and bear in mind when we're doing our work. All right, so I'm going to talk a bit about mainly the stocking now for the rest of the presentation, because that's sort of taken up the bulk of my time, probably over the last 3 or 4 years. I'm only going to really briefly talk about this slide. I'll talk about it in more detail in other presentations. So basically, our entire stocking program is guided by what we call a fisheries management strategy, which is basically a guiding document that we're effectively legally bound to follow. It sets out what species we can stock, what estuaries we can stock, how many fish we can put in a particular estuary, basically any kind of aspect, how we manage our brood fish. All that is written out in this document, and we must follow it, okay? It's law for us to follow that. It's a pretty conservative document, which is a good thing. But yeah, that basically guides you. It stops us from doing rogue kind of stuff, like putting barramundi in Sydney Harbour or something like that. So we couldn't do that. This wouldn't allow us to. All right, I'm going to talk a little bit about recruitment limitation because it's really, I guess, a fundamental aspect of our stocking program. It sort of underpins the work that we do. So it's a bit of theory, if you like, here. And essentially, the recruitment limitation is basically a bit of a mismatch, if you like, between the carrying capacity of a particular system and then the number of natural recruits that are moving into that kind of system. Okay, so we have basically, if you like an excess of resources within a system due to this lack of recruitment in, and when I say system, I'm talking about estuaries here, a lack of movement of fish or recruitment of fish into that particular system. So why might this sort of mismatch occur? This is probably the big one here, which is just a decrease in spawning stock. Okay, so you have just a decreased number of fish just through fishing pressure, which we have on all of our stocks. So the spawning stock is decreased. The number of recruits which would otherwise be recruiting into some of these systems is lower than would otherwise naturally be the case. Adverse advection by the EAC. EAC is East Australian Current, which I sort of try to show in this little map here. A lot of our species in Australia, similar to here in South Africa, have a juvenile marine phase. If those larvae, which are sort of floating around out there in the ocean and depend upon an estuary for nursery grounds, are sort of washed out to sea, then they're effectively kind of lost from the estuary. And we've had some colleagues do some pretty interesting work mapping how these currents move larvae away from the coast, not for the species that we're stocking, but for other species, and demonstrating, I guess, this effect. We have a lot of icols or closed mouths, closed estuaries in Australia. Again, just like you do here in the Eastern Cape, we call them icols. You guys, just stands for intermittently closed and open lakes and lagoons. It's a mouthful. You call them toques here in South Africa, temporary open closed estuary. So same thing, but again, obviously when that mouth of those estuaries are closed, there's a physical barrier to that estuary, so the larvae simply can't, well, effectively they can't recruit into that system. Or there's a combination of these factors. So effectively what we have, say, a carrying capacity of a system, which I sort of show in a sort of schematic here. The wild fish recruit is lower than would otherwise normally be the case. So we have essentially this sort of spare carrying capacity that we can utilize and try and enhance and increase fishing opportunities for our anglers in those systems. So we stock fish, we put fish into those systems, and we make use of some of this carrying capacity. And notice that blue bar doesn't come anywhere near the height of this green one. And that's because there's a whole lot of unknowns there. Yeah, like we don't actually know what the true carrying capacity of the system is. We can model it and we can estimate it. We don't really know the true number of wild fish in every single estuary. Okay, we can't possibly know that. But again, we can kind of estimate it. So we take a really conservative approach and only stock a small number of, or a small portion, if you like, of what we estimate to be the available resources within that system. So as I said, we're stocking 2 species at the moment. The dusky flathead is one of them. It's a bit of a funny species for people who don't come from like Australia. It's like Australians just absolutely just go bananas for these things. They grow really fast. They're easy to catch. They get pretty big. They get to about 1.2 meters. So that, you know, there's sort of trophy-sized fish you can catch, and they're a great table fish. Okay, so they're highly prized species within the rec fishing community in New South Wales. So we're stocking 3 estuaries at the moment: Burrell Lake, Conjola, and St George's Basin. We collect wild brood stock each year. So during the spawning season, we go out, we collect between 30 and 60 brood fish. We use them in our breeding program. The fish come from a couple of different estuaries normally. And we then stock them into these three systems after we spawn them. So I mentioned earlier that we sort of, we do some of this sort of telemetry work. So here's a bit of a case study, an example of where we've done some of that telemetry. So this is like an actual science project that we're doing, not just sort of talking about the program in general. So actually we grew 30 of these fish up in the hatchery, up to a size that we could put a tag into them, an acoustic transmitter, which are these little ones here. And we released them into an estuary called Lake Macquarie, which is actually a huge estuary. It's just south of Port Stephens where we live. We let them go at that little star there. And these sort of blue triangles where we had some listening stations that could detect the presence of these fish throughout the lake. So we looked at their broad scale movements as well as their fine scale habitat use. So out of the 30 fish we stocked, we found 6 of them sort of moved away from that release station. It doesn't sound like very many, but sort of a reasonable portion of the numbers of fish we put in. This is also a massive lake. We didn't really have probably enough receivers, but it demonstrated that some of the fish are going to move as we suspected they would. If we zoom right in now to where we released the fish, we had a whole bunch of other receivers here set up basically to allow us not only to detect the presence of fish, but determine, like, pinpoint roughly or pretty accurately where they were. So over a few months, we I guess able to detect fish in this. I just put up about 4,500 positions just now. So these little purple dots are where we got positions of these dusky flathead over that time. And we found there's a really close affinity between the seagrass and sand edge. So there was a really strong, like, edge effect there. It's not really surprising. Intuitively, you'd probably think that the fish may hang in those kind of areas, but we're able to demonstrate that through this telemetry work that we did. So our stocking program now with these flathead, we typically go and look for areas where we have like a mosaic of seagrass and sand, and that's where we tend to target our stocking. So we don't just take the fish out to an estuary and release them at a boat ramp or from the shore. We'll ferry them out in a boat, in tanks on a boat, specifically into these kind of habitats, and release them into those spots. So the other species we're stocking, as I said, was mulloway or cob to you folk. We didn't need to do any work looking at the juvenile habitat use of these guys because they're pretty well established. We've done a lot of work on this in the past. There's been a lot of work coming out of South Africa looking at the same kind of thing. So we knew where to release these fish. And our work for our stocking for this species is mostly done just south of Sydney, on the southern outskirts of Sydney. And we had to overcome some pretty technical problems for these guys. Which really comes down to the production in the hatchery. So unlike the flathead where we can go and catch 30 to 60, wild fish in a spawning condition, and we actually strip spawn them. It's really easy. It's not a problem. For these guys, it's not possible. We can't just go and catch that many fish for a start in a large, spawning species or spawning individuals. And if you strip spawn them, they'll die for a start. 100% they'll die. And also you have really low fertilization rates as well using that approach. So what we do is we tank spawn them. The problem is when you put them in a tank, it takes at least 3 years before they'll come into a spawning condition. So as you can imagine, that's a lot of time and effort and money to hold a fish of these kind of sizes in those tanks. The other problem you have here is with a tank spawning event, you have no control over the crosses, and almost inevitably you get dominance happening in these systems. So you have one female or perhaps two females will be dominant and will cross with only one or two other males. Okay, so all of a sudden you have these pretty low levels of family variance coming out of your breeding program, which reduces your effective population size and it reduces the overall fitness of the population, essentially. So we need to monitor this kind of stuff, and these are the kind of questions and issues that we're monitoring as part of our genetic work on these species. And yeah, we release them about 40 to 70 mil in size. I might skip over this one because I'm probably running out of time. But we did do some tagging work. Every now and again, we release some big ones. So sort of this sort of size, like 50 centimeters or so. We put external tags in these guys and we let them go and we track them where they go. But I'm probably going to go over time, so I'm not going into too much detail now. But again, genetics, I sort of talk about this ad nauseum because this is probably this is the big one that we need to be concerned about with any kind of captive breeding program. You need to be aware of these kind of genetic implications. And the two key issues, essentially, the fish need to match the receiving estuaries. Okay, so remembering we're taking broodstock from one system, spawning them, and then releasing fish into other estuaries, so multiple estuaries. So you need to have a good handle on the genetic stock structure of that species in the wild to ensure that the population you're bringing your brood fish out of is the same as where you're going to be releasing the offspring of those fish. Okay, they need to match. You're not going to be sort of introducing like deleterious genes into a different gene pool. And avoiding reducing genetic diversity, again, ensuring that you have a fairly high level of effective population size coming out of the hatchery. And you can achieve this by having plenty of brood fish, rotating them over time, all of those kind of things. And we look at all this kind of stuff as part of this program. All right, this is my last topic. I'm just almost finished now, but back onto the artificial reef. So I sort of talked about stocking a little bit. This is a recent study we published at the start of the year looking at interactions between white sharks and artificial reefs. So artificial reefs are obviously designed to aggregate species that people like to fish for and provide a location for people to go fishing at. Sometimes the reefs tend to attract species that we don't necessarily want around them. In particular, of concern is threatened, endangered, or protected species, so TEP species. And the one we focused on was white sharks. Like I'm not really particularly interested in white sharks. It's not really something that really sort of I'm not that into. But it just so happens that like a whole bunch of people got eaten by or killed by white sharks a few years ago. The government kind of had a real strong reaction to that in about 2016 and put huge amounts of money into research programs and monitoring programs of this species in New South Wales. Like millions of dollars was poured into it, and they started tagging these things like that was going out of fashion. There's now hundreds of these guys swimming around the east coast of Australia with a tag, but there's probably not many of them without a tag, to be perfectly honest with you. I'm not joking. Like they've tagged 500 or 600 of the things, maybe 700 of the things, so lots of them. The tags they put on the sharks are able to be detected by the listening stations that we happen to put on our artificial reefs. So we're able to monitor quite easily, which is why I focused on white sharks, interactions between this species and our artificial reefs. So we had a look at basically 7 different reefs, which I've got listed there. They're kind of listed in like a latitudinal sort of sense, from Port Mac up in the north down to Marimba in the south. And we monitored them between late 2017 through to 2021. The grey shaded areas there show when we monitored them. It varies from reef to reef because they went in at different times and we monitored them over different periods, so it wasn't perfect. But during that time, we found there was 590 visits of white sharks to these artificial reefs from 199 different sharks. Okay, so quite a variety of individuals were coming up on our reefs. We found the timing of the interactions tended to line up with their seasonal migrations that we know about, because pouring all this money into this stuff. We know a lot about this species now. The interactions tended to be short. So generally only about 14 minutes or so was the mean amount of time I spent there. So really they were just sort of coming on in, having a bit of a look-see, and then moving on again. I think the longest interaction only lasted about 3.5 hours. And these are actually kernel density plots. They're kind of tilted on their side, which sort of shows, and this is the scale here, but this shows it was really short. Generally, they were quite short. We found the time they spent at the reef was slightly affected by chlorophyll A. So we did some modelling on this, with a positive effect of chlorophyll A and a negative effect of shark size. So bigger sharks spent less amount of time there. But it was really only a matter of minutes. So it was kind of, yeah. It was significantly statistically, if you like, but really kind of not that meaningful. But I guess the neat thing about it was not only did we show this, but it was also, I guess, a demonstration of a way in which, if you were interested in looking at endangered species and their interactions with things such as artificial reefs, these approaches worked kind of in a pretty neat way. And we were able to piggyback on this shark work, and it was kind of lucky it worked out for us in that way. And I'm going to stop there because I've probably gone for long enough already, but there's a whole heap of people who are involved in all of this work. This sort of stuff, as I said, has been going on for quite a while now. So inevitably, lots of people have been assisting in it. So thanks. Might just end that show.
Thanks so much, Al. Any question for Al from the audience? And then we'll see if there's anything from online on the chat. Yes, one question here.
Yeah, thanks, Alistair. I think very interesting. Do you have any indices of what portion of your stock fish are killed by anglers?
Yeah, it varies amongst those species. Yeah. I didn't say that on purpose, waiting for the question, basically. But for the prawns, for example, we had, depending on the system, some of them, every prawn they caught in that system was stopped. Yeah. With the kingfish, no, we actually tracked them acoustically as well, and they just vanished. And we stopped that program basically because of that. For the Mulloway and the dusky flathead, some of the earlier work that we did showed that about 7 to 12% of the catches within the estuary were stopped. Yeah. More recently, it's been far fewer. We've had, with that FMS I talked about, the fisheries management strategy, placed a bunch of restrictions on our stocking program. So more recently, yeah, the returns have been much lower. So for example, we've sequenced recently 400, I think it was 400 samples of Mulloway, 300 samples of Mulloway for one. Okay, but that's also on the back of probably been a mass natural recruitment. We had a lot of wet years in Australia. So we had 3 or 4 really wet years in Australia, which has led to like an absolute boom in natural recruitment, which is obviously, it's a good thing. But it's probably dwarfed, if you like, our stocking program. So yeah, the results vary. We're still pretty early on in that stocking program. There's some things like the change, the size of the fish that we're stocking, a bunch of things like that. But yeah, at the moment, the most recent stuff impact has been low, but again, it depends on the species.
Yeah, and then I was just to follow up, what, in terms of the money that comes in for recreational angling, what portion of the money is spent on this kind of work compared to other recreational fisheries work?
That's a really good question, though. Something which has been put at politicians in the last couple of weeks, actually. It's hard to put a figure on it. The money funds a whole bunch of things. Okay, putting in perspective, the revenue is about 17 million Australian dollars a year. Those reefs cost about 2 million each, and they're putting them in on an annual basis. The stocking program costs about $1.5 million to run, so that sort of gives you a bit of a figure in terms of that. They fund a whole bunch of other things. They put in fish cleaning tables, fishing structures, but a lot of money does go to paying like managers' wages and stuff. And there's questions about how much of that money should be allocated to basically government salaries versus on-ground works. So that kind of maybe answers your question. I can't give you a figure in a percentage, but that's gives you a bit of an idea, I hope.
So it's just quite interesting to see how budgets are.
When that money was first, when they first put in the license, it was basically sold because bringing a license is obviously difficult. Hey, suddenly so basically tax people, if you like, to go fishing. It was difficult, but they sold it in the first few years of that license fees. They bought out the commercial fishers fishing in a whole bunch of estuaries. That's what the money went to solely. So we now have these rec fishing havens, as we call them, where there is no recreational fishing permitted. And that was, they had to buy out those commercial fishermen in those systems. And that was when their license was first brought in. There's just some talk that they should buy out some more. But again, it becomes political and difficult. Remember, we're a government body still. Nothing happens quickly or dramatically. That's for sure.
There's one question from Alan and then one from Russell.
Amber's work on plastic carbon Algo Bay showed that there were essentially two subpopulations, if you want to call them that, a more marine one and a more estuarine one. Now, in your stocking program, you said that you collected your fish mainly from the estuaries, the adults that you were going to use. But if, let's say, you indicated that there was a dominance by, let's say, one or two females. If one or two of those females, let's say, had been a marine mulloway, when you stock those offspring, they wouldn't end up going into the estuary. They would then colonize the marine, adjacent marine environment. So then you would get an expected low return from your anglers of stocked fish, because those youngsters are a marine mulloway stock and not an estuarine mulloway stock. Do you have any genetic evidence to suggest that your mulloway do comprise two separate stocks?
Not at this stage, but we're just in the process of starting a new federally funded project where we are going to look at the genetics of mulloway in a lot more detail than we have before, using like genomics. So in the past, we're based in the work that was published, I don't know, probably 15 or more years ago now, which had multiple samples along the New South Wales coast, which showed like a panmictic population, and that is what we're sort of based our stocking on at the moment. We collect the mulloway from the estuaries purely. It's easier for a start. We've got to try and transport these things. They're difficult. They're a soft fish, like they're really soft fish. Bringing them up, we could beach fish for them. It's another option we have thought about. But fishing from a boat, even in waters 10 meters deep, we've been shown to have barotrauma. So even from 5 meters down, they can get barotrauma. So yeah, it's a difficult one, like a management of those brood fish. But we are, as I said, about to embark on a new whole genome work on mulloway. You get a really good handle of some of those. Yeah, those I guess they're sort of real. On the camera, those sort of fine scale, like genetic kind of questions about that stock for sure. And there's a whole bunch of other things about whether, certain fish are more, I guess, adapted to spawning in tanks, and whether those fish are so suited to the wild. There's a whole heap of different genetic questions you can look at there. Yeah.
Thank you. We get Russell, and then we hold on, Matthew. We've seen you, and we'll unmute you just now. But first, Russell, thanks.
So I just want to ask, you mentioned something. There's a catch monitoring program, integrating anglers with the catching, etc.
Yeah. Yep. It's Yeah, it's
So are you able, have you been able to track a change or increase in the production of the fishery through that program or not?
Yeah, no, so we're not getting effort data, unfortunately, yet. So that we're not, it's when the citizen science program is more about those anglers just providing us tissue samples. We're not, say, diarising them to get information on sort of catch rates and how that might work over time. We've tried. We have, like, for example, like a phone app, which the department has put out, and I've tried a lot to get anglers to use that app and record their catch. There's obviously massive biases with that, where anglers are far more likely to record fishing trips where they catch something compared to when they don't catch anything at all. So there's a whole bunch of issues with that. We do have a broader monitoring program within the state. It's a phone diarised, so they interview about 1,200 fishermen over a year. It's probably still, I mean, the chances of detecting those kind of changes, and it's not really, it doesn't drill down into like an estuary kind of level. Yeah.
You don't have a dedicated like enumerated site collecting data on the catch statistics.
Not in that, no.
Thank you. And Matthew, can you just unmute yourself and ask the question, please?
Hi, thanks very much. Can everybody hear me?
Yes.
Yep.
Okay, great. How's it, Alistair? Thank you so much for that talk. I've got a double-barreled sort of question I wouldn't mind getting your insight on. So obviously, it's sort of widely accepted that carp in South Africa are in a pretty poor state, and there seems to be a growing pressure from perhaps the rec angling community and others to consider stocking as a solution. My question to you is, where in the spectrum from sort of infant level fisheries management to gold standard fisheries management, where in that spectrum do you think stocking enters the equation? And then my next sort of extension of that question would be, and then how do you manage angler expectations and behavior change once you've started stocking fish?
Yeah, so the expectation of the anglers angler expectation is an interesting one. We certainly attempted to temper expectations. We don't, the stocking program isn't there to massively alter like our fisheries. As I said, it's like an extra tool in the toolbox to the work that we are doing. I've been out in the field. We're doing our work, and we have anglers coming up to saying, "Oh, yeah, I've caught all these, these massive flathead. The stocking work's going fantastic." Which my answer is like, there's no way there's stocked fish. They're too old to be anything that we've stocked. We obviously we promote it as we would, because we're using fishing license fees to do the work. But yeah, it's not sold as something which is going to like radically change the recreational fishery. As I said, most of the fisheries management actions are done through those more sort of traditional types of approaches, those input controls, output controls. We've recently reduced bag limits on Mulloway, so anglers can now only take one fish a day. So I mean, it's about cut as much as what we can do. Other than that, you're looking at basically a moratorium on that. There's been restrictions to the commercial catch as well. I know I'm sort of getting off track a little bit here, but yeah, essentially, yeah, we don't sell the stocking program as something which is going to massively radically change their fishing experiences, but hopefully enhance it to a degree. And there was another question there which I kind of missed at the beginning. I'm not quite sure what it was, but.
Do you want to repeat it, the first part, Matthew, or you happy with?
Yeah, I think you've answered most of it, Alistair. The question was really just, I suppose it was kind of hinting at the idea that in many places it's seen as a silver bullet to recovering stocks, if you know what I mean. And I guess my question was just, where does it enter the equation from that point of view? And
yeah, it's certainly. I mean, look, the first thing I would say to anybody,
really. So
yeah,
I think you've answered that.
Yeah, I mean, that's the first, I guess, response to that is it's not a silver bullet. Like, absolutely, it's not. For example, the work that Vicky will talk about in a minute, looking at habitat restoration. I mean, there's no point in stocking Mulloway into a system that doesn't have the supporting habitat, for example. I mean, you're just wasting your time. There's no point in putting mulloway into a system where they're going to get scooped up in a trawl fishery, like an estuarine trawl fishery, you know. So it's only part of the, you know, like an extra tool in that toolbox. So it comes in, I guess, I would say that the lower end of the spectrum in terms of our management actions. It's certainly not at the top, that's for sure. If that answers the question, I think it probably does. Yeah.
Thank you so much. One last round of applause for Al. Sorry. Thanks, Al. And now it gives me really great pleasure to introduce the second speaker, Vicky Cole. Vicky was postdocing here with us a few years back, and now she's a research scientist based at the new name that I can read now, the Department of Primary Industries and Regional Development, Fisheries and Forestry. And she's going to tell us more about oyster restoration. So the IMISI project in the house, open your ears and ask questions. Thank you, Vicky. I think it should be okay. Try like that, because I tried it here. Should work. Yeah, thank you so much.
Thank you. So a bit of a change of things. I'm going to be talking about oyster reefs. So basically this is a talk on behalf of all my co-authors who are the oyster reef team at DPI. So oyster reefs, such as you find with all shellfish reefs, like mussels here, they're really important habitat for biodiversity and ecosystem services they provide, such as filtration. Oyster reefs were once really abundant in New South Wales and really widespread. It's kind of thing that we've seen throughout the world, but unfortunately, they've been lost due to overharvesting and disease and declining water quality. So in 2018, the New South Wales government put together this New South Wales Marine Estate Management Strategy. And it was a whole vision to have a healthy coast from now into the future. And as part of that, us in the oyster reef team plan to develop and deliver innovative projects to fill knowledge gaps and restore oyster reefs, securing the future of oyster reefs in the state. So I guess we started when we realised that pretty much we didn't know much about oyster reefs. And it's kind of a forgotten history. So what we want to know is where were the oyster reefs, how many there were, and how they were lost. So we employed this environmental historian, and she found these really interesting things. So she found, like, back in the day, like pre-European settlement, the oysters really abundant, and also, you know, they had so many that they formed these massive middens they could use for lime, they use for food, but unfortunately they're all lost very quickly. So in a quite short amount of time, you would not see many oyster reefs. And that she also found, you know, various places had names like Oyster Cove and Limeburners Creek, things like that. And they're kind of evidence that these are once really important places for oyster reefs back in the day. So with this loss, it was also a really big impact on Aboriginal people. So these oyster reefs and all shellfish reefs have really significant cultural importance to them. And communities used to gather around shellfish reefs. They're able to sustainably harvest them for many thousands of years. And in some areas, they actually worked them and were able to harvest them as well. So we've collaborated with university, trying to work out this, like, I guess, looking back even further in time, thousands of years. That's what I'm doing there. Another thing we realised, we actually don't know where the oyster reefs are now, so we're mapping them. We're doing this through aerial imagery, and then going in the field and ground truthing it. So at the moment, we've mapped about 45 estuaries, but given there's 145 estuaries in New South Wales. It's going to take a while, but you can check out the data on this website so everyone can access it for free. What we were trying to do, I guess, in this whole vacuum of no knowledge about oyster reefs is to build this idea of social license to try and encourage people to accept and be on board the whole oyster reef restoration program. And we did that through targeted surveys, engagement and outreach. So we had infographics, we attended public talks, went on river paddles, attended lots of different events, have social media, newsletter articles, and we've also got a travelling museum display. So we have a travelling museum display called Ginger, which means oyster in the language around Sydney. So basically it's a collaboration with the Australian Maritime Museum. And we're, I guess, taking the story of lost oyster reefs and the restoration to regional coastal towns. And the good thing is we're able to, I guess, bring it to different places and have talks and things associated with it. And I guess this whole display, as you can see in these pictures, it reflects this strong acknowledgement of sea country. So I guess the Aboriginal link to sea country. So in this map, you've probably seen the ones from but I guess that's, East Coast of Australia, where we are in Port Stephens, where we are in relation to Sydney, which is about 2.5 hours north. And we've got oyster reefs restoration sites in Port Stephens, Botany near Sydney, and Morgonga down south. So we first started the first oyster reef restoration project in Port Stephens. And it now is about 11 soccer pitches in size, so quite large. And we're doing this restoration mainly as part of the government, but we also collaborate with local councils and the Nature Conservancy and various other federal and state agencies as well. And we're doing restoration of just not the Sydney rock oysters, but also the subtidal and Gazi reefs. So they're both native oyster reefs. So I guess my job is, as a scientist, to design a whole program in order to detect environmental change of the oyster reef restoration program. So I guess we look up here, I guess when we have bare sediment, so what we started off with, and then we sample for approximately a year beforehand. And then now we're kind of in that red section. So how long is it going to take for the bare sand to become more similar to a natural oyster reef? And so that's there. And I guess our whole aim is to experimentally test if oyster reef restoration is successful for invertebrates, fish, and their overall ecological functioning. And we did that by doing various different things, but it's just basically looking at the diversity, abundance, and size of fish, invertebrates, and oysters themselves. And in doing that, we have various new technologies to do with eDNA, using drones, and using AI. So we've also got a fair bit of extra help due to citizen scientists. And they've been helping us with counting the oysters themselves, mainly down south. So it's about a 9-hour drive for us to go and sample there. So with these citizen scientists, they can do it more frequently than we'd be able to get down the south coast. And we also have a group of Aboriginal divers who help with monitoring the subtidal oyster reefs as well. So what the program was, so before the on-ground works, we're mostly interested in understanding this natural variability in assemblages of oyster reefs and bare sand. So the bare sand, we had like quite strong differences between the bare sand and the oyster reefs, and that's just naturally. So on the right, you can see the control and the restoration. So that's just bare sand. So the red and the dark blue. And that was significantly different from the light blue, which are the reference sites, which are oyster reefs. That's in terms of the invertebrates in Port Stephens. We also found the same with fish. So we found more, we had different assemblages, but also we found more fish in the oyster reefs compared to bare sand. And that was detected with BRUVs and also with eDNA. All right, so then we can start measuring success of the program. I guess that's where things start getting a bit more exciting. So as part of that, we had a chance to test different types of, I guess, rock bases, different reef bases, sorry, which are either rock or shell, and deciding which one was more important. So we did this in Port Stephens. So we had an asymmetrical design where we had two, I can't really see it very well, at the two reefs. The first one is in Gura, which is sea country, and the one at the bottom is Bindama, which is called black possum sea oysters. And there were two reefs, which are made of rock, and another reef, which is made of rock and shell. So as part of that, we're able to test which one's better. And that will kind of guide whether we're going to use rock and shell in the future. All right, and it was pretty clear. We found really strong differences with the rock and the shell. So the rock and shell, the combination of the two, is a dark blue. And for oysters, we found a lot less on the rock and shell than rock by themselves. So I guess, this is quite different to what's found overseas. So most overseas, they use shell, and they found that most oysters settle on shell. They like conspecifics. But I guess not too surprisingly, when you think about, they're called Sydney rock oysters, and they prefer rock. So that's what we found for rock and shell. And this is we also found for Bembesim as well. So we found a lot more on the rock than the rock and shell. In terms of fish, so this is for brim, we found no real difference. So yeah, it might take more time, but probably not, don't care. So yeah, I guess this is kind of one way we were able to change how we do oyster restoration. So when we went to expand the reef, then we only used rock. And then when we use the new reef down south, it's only rock as well. All right, so how's our restoration progressing so far? I guess because Port Stephens was the first one, I'm really going to focus on the results of Port Stephens. And we also produced these infographics of how it's going. I guess there's a few important points. We've got over 34 million oysters that have naturally recruited to the restoration site. The oysters themselves are now about 80% of the size of natural oysters. And we've got about 35 different species on the new reef, which is similar to what we see on natural oyster reefs as well. And this is just what they look like at the subtype 1. These are quite new, so this is only a year to 2 years old. All right, so in Port Stephens, in terms of the fish, so you can start seeing a bit of a difference. So red are the controls, the bare sand, dark blue are the restoration, kind of in the middle, and light blue are the oyster reefs. So you can see we're starting to see the restoration ones that are moving across to be more similar to natural oyster reefs and less similar to the ones in red in the bare sand. So that's a good sign. In a bit more detail, These are oyster blennies. So you can see they're slight increase. So they're getting there, but not quite. Other species, such as bream, have actually overshot the mark. They're way more abundant on the restoration site than the natural reefs. Other species, such as the whiting, have actually decreased, but that's what we expected. You find more whiting on bare sand than on oyster reefs. And other species, such as gobies, they've actually yet to respond to the oysters and the restoration. Right, and other species, such as the ludwig, they've actually increased, kind of similar to what we saw with the bream. And that's the same for intertidal Sydney rock oysters and also subtidal Angazi reefs. In terms of the invertebrates of Port Stephens, they've also increased, you can see in the dark blue. Again, it's still got a bit more to go to be similar to the natural oyster reefs. So I guess similar to the blennies. All right, and I guess, you know, it's not just the biodiversity, we're actually seeing increases in terms of ecosystem services. So this in terms of the filtration of the oysters. So here we've got this little experiment where we put these, I guess, containers over oyster reefs, and we can measure the filtration. So what we found is the 2021, I guess, of the new reefs, 2020 are the original oyster reefs that we restored, and oyster reef is just the natural ones. So I guess the old ones, they're kind of getting there, but not quite in terms of the filtration, and the new ones still have a bit more to go. But with these numbers, I was then able to calculate, based on the area, approximately how much area and how much filtration was being done as part of our restoration. So what we worked out was the oysters that we've restored, or the reef that we've restored, are now contributing about 9 million litres of water per hour of filtration. And that's similar to about 3.5 Olympic swimming pools. So every hour they're filtering that much water. We've also been doing some environmental economics. I guess that's just with the Port Stephens reefs, because that's been the most one that's been going the longest, and that's been economists at GPI. So what he's doing is looking at the cost-benefit analysis of doing this quite expensive restoration and the benefits that we're getting out of it. So I guess the costs are the rock and I guess my time, and the benefits of things like improved water quality, recreational fishing, carbon sequestration, shoreline protection, and I guess other non-use values as well. And what we've found is the benefits are highly outweighing the costs. So probably about, I think it's 7 to 1. So really, really good sign that this kind of work is beneficial in a dollar way. Another thing we've been doing is not just focusing on Sydney rock and Angazi reefs, we've also been looking at other species of oyster reefs, other species of oysters. We've been collaborating with the uni, and we've been looking at species that I guess not well known, and these oysters can live in really poor quality spaces. So I guess that's one of the things we can't do at the moment, is only do restoration in relatively clean places. So, you know, maybe some of the things that we want to happen can only be done in nice places anyway. So this is, I guess, a potentially useful oyster that we can use, and a potential species for restoration in the future. So just a bit of summary of what I've talked about. So the pre-restoration assemblages varied over time and different among estuaries. Didn't really get into that, but I guess importantly, the oyster reefs had really different assemblages to the natural sediment. and post our on-ground works. We found that rock was a lot better than shell for Sydney rocks. Some of those were starting to become more similar to the natural ones, which we saw for the fish. And we're also starting to see some ecosystem services, such as the filtration, starting to appear. So I guess in the future, we're going to continue this monitoring. Also, with the successful seeding of the agassiz, going to continue the same thing as well. So I guess we're starting to see these other things we can start to measure, which is how the filtration works of the other estuaries and the agassiz. We're also doing another experiment. We're looking at the usage of habitat by fish. So that's an experiment I've been doing with the VPS around oyster reefs and the restoration site. I guess all in all, it's all looking pretty promising for oyster reef restoration and oyster reefs in New South Wales. So please stay tuned for exciting results to come. And you can do that by our website. And yeah, like to thank all these people and the different communities where oyster reefs are. Thank you.
Thanks so much. Thanks so much, Vicky. Very inspiring and great to see how restoration programs can work. Any question for Vicky from the audience in the room? Alan.
The establishment of a new oyster bed and the complexities that a potential natural oyster bed, which could be decades old, are we not looking at a very short time frame here, which sort of less than 5 years, whereas maybe you need to look at a longer time frame, because as the bed develops, as you said, those oysters are cleaning the water, so the water turbidity is dropping as you go from 1 year to the next, which is changing the ecosystem in a way which you can't really measure in the early stages, because you've still got high turbidity, let's say, because the oysters aren't coping with the turbidity. So what I'm trying to say is the complexity really only going to be fully understood further down the line? In other words, we're looking at a very early succession stage as opposed to a mature oyster bed.
Yeah, so definitely. And this program goes for 10 years at the moment. So we're only in, I guess, the 6th year now. So I guess at the moment, the plan is to measure these for 10 years. And then hopefully after that, we'll probably not be able to measure, do the same thing as often, but we're definitely keen to keep measuring because, as you said, we are going to get change later on. And that's kind of the problem with all these restoration projects. A lot of them, the funding only goes for a very short time and we don't see change. And this is just, this is not hinted what's happening so far. But what they've found with, I think it's SAIAB, things are actually, they look good at first and then they start decreasing. So, this is something we need to actually measure for a lot longer. And it's definitely, So at the moment, 10 years, but there is definitely plans to a lot longer after that as well.
Yes, and one
Thanks,
question.
Vicky. I was wondering about the volunteer programs that you have going there, and did you see any useful data coming out of that? And would they potentially be a solution to that long-term monitoring?
Yeah, definitely. So especially the ones that have been helping down south. So they've been, I guess the first thing was to sample more frequently than us. And they're also sampling longer. So they've only sampled for 1 year at the moment, and they've already signed on to sample for another year. So definitely we'd like them to continue to do that. Yes, it's I guess making sure the enthusiasm stays for years to come.
I see that there's no question online, but I've got a question for you. Comparing to some rehabilitation work, coastal work that was started in this country, and how giving access to people has kind of reversed the good work of the rehabilitation. Have you considered anything in terms of giving access to people onto those restored reef? And if yes, is there any, is there any impact at such a short time frame?
Yep, so at the moment, all our reefs are open to fishing. So anyone can go and harvest. I guess there isn't much of a need to harvest oysters themselves at these sites. But in terms of fishing, we do encourage them, like that, you know, people are keen to fish and we want them to, because if they can be supportive of the oyster reef, they're going to be supportive of the restoration. And I guess that's why we've been doing this project with the VPS to actually prove the oyster reefs are beneficial to fishers. Yeah.
Thank you so much. Any more questions for Vicky? I see there is none online, but any? Yes, Alan, please.
I don't know if you, this is a difficult one, perhaps. In South Africa, we've got oysters along the coast, and sometimes, or were, very abundant. But in the estuaries, you don't find that. Even in rocky areas, there's never, it sounds like the estuaries in the east coast of Australia, which is not dissimilar to our east coast.
Yes.
And our estuaries are not dissimilar to your estuaries. And yet we don't have large oyster beds, although we know from Nasda Estuary, for example, that oysters can survive in the estuary if you provide them with a means to be there.
Yeah.
So why has South African estuaries got so few oysters?
Yeah, I don't know. They're like they're really similar. Like even, I guess, open coast, like, you know, your shores are covered in mussels. We don't have mussels on our shores. How they all? Yeah, they're really different, and I'm not sure. Yeah, I guess that's the species that's using that niche, and that, yeah.
Can I ask about maybe wave dynamics? So I don't know if I've just visited some of your oyster restoration site, and it looked pretty much sheltered, and maybe in estuaries, I don't know, Alan, there's still quite a bit of reflection of the quite high coastal wave dynamics that maybe can translate into it's somehow a reflection of that way of the strong hydrodynamic system that the rocky shores are, and that's why mussels are doing very well here in terms of exposure to high energy wave. So maybe there's some sort of link to the patterns of the water hydrodynamics at the two different systems. But maybe I saw the site at a very nice.
I think you got really nice days. Plus even is a massive estuary, and it gets really rough.
Okay.
Yeah.
Well, if there's no more question, I really like to thank you for this afternoon talks and series. And I would like especially to thank Al and Vicky for their time during the trip to South Africa. Thank you very much. And I thank all of the online audience. Thank you. And to the next one.